good afternoon, everyone. So my name is Niels Geudens, and I'm a data steward working at VIB or Elixir Belgium. Although I understand from the previous talks, I might change my title to Institutional Data Manager, but okay, what's in a name? But thank you for the useful insight either way. So to give you an idea of what's to come, I'll first introduce who we are, and then I'll start talking about data repositories for fair data. And you'll start to notice that a lot of my figures or slides were actually partly already presented in the last presentations. But that's great because then we have a very strong foundation to build further on our DataHub platform, which aims to kind of deliver fair data by design. Okay, so first let's start with who we are. And first I'd like to introduce Elixir, which is an intergovernmental organization that brings together life science resources from across entire Europe. Its main goal is to create a unified and accessible infrastructure that allows researchers to discover, access, and work with life science data. And these efforts help simplify the things that researchers may struggle with, such as, for example, finding the right data sets or understanding how it was generated or shared their own data in a very standardized way. And just to mention, Elixir is also a formal partner of Euro-BioImaging. Then zooming in from the European level, let's first look at Elixir Belgium, which is our national node within the Elixir infrastructure. And the Belgian node is made up of 9 partners, which include all the Flemish universities, 2 Leuven universities, Cienzano, which is the Belgian Institute for Public Health, and then VIB, which also leads the node, which is also the institute where I work. So VIB, the national node, and then the group where I work, the Data Core group, aims to support researchers in 3 core areas, which is 1, the software tools and workflows for analysis, hardware and infrastructure, which includes the computing cluster and data storage services, and then finally the part where, or the team where I work, which is the data management, where we offer platforms, training, and guidance for making data structured, searchable, and reusable. Okay, that's pretty big introduction. So let's move on to the core topic of my talk, which is how we aim to manage, to structure, and to share imaging data. Although imaging data, we're not limited to imaging data alone, but of course, considering the audience today, I did make it a bit focused on imaging. So in this first section, we'll first focus on data repositories and how they support the FAIR principles in practice. And then we'll also touch upon some of the difficulties or the challenges that researchers face in getting data into these repositories and how we then aim to help with this. So these are things that we already saw in the previous talks. So how do researchers actually identify and choose the suitable repositories for imaging data? Well, we already learned about this in the previous talk right before lunch, and I just want to highlight FAIR sharing as a key resources here. For example, you can visit the catalog of repositories, metadata standards, and policies that are specifically recommended and supported by Euro-BioImaging, and then also the decision tree that we saw in the previous talk. Now, 2 prominent repositories that I want to highlight here are Bioimage Archive and Image Data Resources, or IDR. For the remainder of my talk, I'll probably mostly talk about Bioimage Archive and its REMBI metadata model. However, it's also worth noting that IDR plays an important role because it emphasizes the highly annotated reference datasets, which we're also actively exploring in our DataHub, which I'll introduce later on. Then depositing data in the repositories is essential for effective data sharing. But researchers often still encounter some practical hurdles that make this process less straightforward than it should be. And again, a resource that I would like to highlight is the FAIR Cookbook, which is offered by Elixir, and the Euro-BioImaging has created a step-by-step recipe, let's call it then, for data submission in the bioimage archive. And then again, introducing the REMBI model, which we already saw a couple of times, so I can be short. So just to recap, REMBI stands for the recommended metadata for biological images. And it basically defines a minimum and extended metadata needed to describe imaging data sets when you upload them to a repository like, for example, Bioimage Archive. Structure is designed to cover everything from a high-level study design down to the technical acquisition details, for example, of the images itself. And then, so we'll use a REMBI to describe that data that is uploaded to Bioimage Archive. Now, the structured approach also means that metadata fields are not only captured consistently, but also that they are mapped to controlled vocabulary, including, for example, also including license and provenance. And as a result, by including this metadata in a repository like Bioimage Archive, the data that is stored there is immediately ready for support with fair data sharing. For example, data becomes findable because it includes rich metadata that can be indexed and linked to persistent identifiers, can be accessible through standard protocols, interoperable because it adheres to community standards, and reusable because we include the necessary licensing. So basically, fair data. So in essence, REMBI makes sure that data in repositories is fair, so that they have an output that is fair. However, in practice, there are still some major barriers for researchers that are trying to prepare their metadata. For example, there's on one hand metadata complexity, where researchers are expected to navigate multiple standards for different repositories. Then there's also a timing issue. In most cases, into my experience, but let's be optimistic, sometimes too often, metadata is collected post hoc, which means that after the experiments is done, and that leads to important context that can be forgotten or hard to reconstruct. And then finally, there are also some technical barriers, since most researchers work in spreadsheets, lab notebooks, or custom formats. Converting this into a structured metadata which is suitable for submission to the repositories is not always trivial. And so these challenges highlight the need for tools that just don't support FAIR principles, but basically we need to insert them directly into the data creation process. So basically, to guide these challenges, of course, we have a tool developed for that. I'd like to introduce our DataHub platform, which is a tool which is designed to guide researchers through the process of metadata capture in a way that both standards compliant, but also submission ready from the start, so at the time of data creation. And as mentioned, DataHub is a metadata management platform, which is designed to support researchers throughout the life cycle of an experiment. Platform is centered around the idea of capturing structured metadata from the start, so thereby following a FAIR by design approach. But this means that metadata is collected in a way that naturally supports FAIR principles instead of being retrofitted later. And DataHub is also built to comply with the metadata requirements of the public repositories, databases, making the downstream submission process smoother and more consistent. And we also saw this before, so thank you for the introduction, Kathy. DataHub is an instance of FAIRDM-Seek, which is an established open source platform designed for sharing research data and outcomes in a very structured and transparent way. Now, Verdum-seq in itself is widely used in systems biology, and DataHub does use that solid foundation, but there's a difference in the sense that DataHub is specifically optimized for capturing and managing metadata that comes from multi-omics experiments. And so the focus here is to enable standardized metadata collection using the machine actionable ISA-JSON format, similar as the Verdum-seq in ISA, which is then widely accepted in deposition workflows. And so thereby we ensured compliance with the requirements of the major public databases to kind of help streamline the submission. So again, let's talk back to about the REMBI. This outlines the key metadata components needed to describe the bioimaging datasets in a very standardized way. And so what we did here in DataHub is that we mapped the REMBI's components into the ISA-JSON framework. And this means that each of the metadata elements of the REMBI, whether it's specimen preparation, acquisition settings, or the image file descriptions, these can now be captured in structured templates using a standardized format at either the study level or at the assay level, so the S or the A in ISA. So how does it look in DataHub? Well, for the study information, that's what you see here. The figure shows the key fields which are required for the study submission to Bioimage Archive, for example, as included in the DataHub platform. These include elements like study title, description, but also specific fields such as the license, the keyword, and things like that. So this is captured at creation of the study, making sure that they're not forgotten or added in the last minute. And so this helps to reduce the manual back and forth that typically happens in the process when you start with uploading. Then also for the study components itself, you can see here how the templates, the metadata templates look like. Each of these fields in the template include elements that make the data both user-friendly and machine actionable. For example, every metadata attribute is clearly labeled with descriptions and guidance provided to help the researcher understand what is expected. And second, the type of input is defined, whether it's then controlled vocabulary or a link to another sample or free text. This helps them to standardize the metadata that is being captured. So rather than the researcher needing to learn how to structure all the data itself, this is basically handled by the templates behind the scenes. And then also, sorry. Yes. So for each of the experimental steps, then there's a template available that is created by us. But it can be implemented by the users themselves. Now, DataHub organizes these templates within a structured project hierarchy, following then the ISA structure, so the investigation, the study, and the assay. So this is what you see here. I don't know if you can use my pointer. So here under a specific project, you implement an investigation. The investigation contains the overall context of the research project, the goal, collaborators, and so on. And then we start with the templates under the study or the assay level. Each study within the investigation then captures a specific experimental question. Or biological comparison, whether it's to include metadata like species or something like this. Then we also have the assays, which then represent specific data generating activities, for example, the imaging or the sample preparation. I'm running out of time, so I'll go a bit faster. So once then these templates are implemented in the ISA structure, then of course we need to get the actual data or the metadata filled into the DataHub platform. And so the researchers can interact with the templates and provide their metadata through a dynamic table interface, where each column of the table then represents a metadata field. There is a description of what is expected for them. There is also a real-time validation happening. So if there's missing values for mandatory fields, formatting issues, or some things like that. There are also ontology terms that show up in a drop-down menu. So researchers can then select their terms without or search within the list. Sorry. And then also very interesting to highlight is that we can also download the sample template once it's been implemented via the dynamic table. Users can download that and then fill their data in locally, for example, via Excel, which apparently researchers love to do, working with Excel. So basically you submit the local data. Once you're finished or once you're happy with your set of experiments, you upload the sample template to the assay again, and then all the data is imported into DataHub automatically. Of course, in DataHub, metadata templates don't exist in isolation. They're actually part of a connected data flow that mirrors the structure of a real experiment. For example, one sample, such as specimen preparation or an imaging slide, can serve as an input for image acquisition assay, which in turn produces a digital imaging file. So that's the output then, which can then serve as an input for the data analysis template, where you can store other metadata then. This also supports more complex scenarios, like one-to-many and many-to-one sample relationships. For example, a single sample might be imaged multiple times under different conditions, or multiple processed data sets may be derived from a single raw data file. And then one of the strengths of the sample linkage, then this by extension of DataHub, is its ability to track and link samples across the different assay types. Something which is then especially valuable in multi-omics studies. So hence we don't focus only on bioimaging, but rather on multi-omics, which include the bioimaging. I've drawn up here a very schematic example where we start, for example, from a study source. So that could be, for example, a cell line. You capture the metadata about the origin of that cell line. Then you culture that cell line under specific conditions, generating a study sample, which is then, well, the subject of two separate experiments from bioimaging and the genomics. Each of those assays, or assay streams rather, capture different types of metadata, which is then tailored to the requirements of the repository it's destined for. In the case of bioimaging, then you do the submission to Bioimaging Archive, for example, and then the one that you did for genomics, you can submit to ENA for sequencing. And the important point is here that both assays are connected back to the same sample and the same source. So sample provenance is maintained across the different omics types. And this enables then the consistent data sharing and supports the downstream integrations, making it much easier to understand how the data sets relate, or even reanalyze them in a multi-omics context. Sorry to go over time, almost finished. And so everything that we've seen so far, from the templates, control vocabularies, and sample linkage feeds into this, which is basically the export of the metadata in the ISA JSON format. This format respects the official ISA JSON schema and structure, which is widely accepted across different deposition databases and the brokering tools. It captures not just what materials were used, but also how they were processed following the classic ISO structure, which is then input, protocol, output. And each of those entities is described in its own context, and that export then reflects the relationship defined in the templates that we use. And so this is what we mean by FAIR by design, which is the machine actionable metadata that follows standards, but built into the research process from the start already. And then as a final slide, the diagram summarizes how DataHub aims to support a flexible standard compliant data submission workflow. Once experimental metadata is captured in DataHub and exported as a ISO-JSON file, it can follow multiple paths, for example, direct submission to a local archive, or if in some cases a conversion is needed for upload to other repositories. And I haven't really talked about this in detail, but okay, DataHub doesn't store the actual data files, only their metadata, but we can provide a link to the file's location, and then can be packaged together before the submission. Well, so then we can submit the data and the metadata together. Future direction, I will skip in the interest of time. And that just leaves me to thank VIB, the Data Core, especially Elixir Belgium, and of course you for your attention.